Hello, and thanks so much for inviting me to speak. I'm going to share some basic knowledge of radiation's impact on health. There are a number of different types of radiation. I will be talking exclusively about ionizing radiation. And I contend that understanding radiation's impact on health has been difficult, not because it is full of uncertainties, which it is, but because radiation science has been plagued with poor assumptions and incomplete damage models. It has failed to follow the obvious and critical questions needed to help us understand and deal with these uncertainties, particularly damage to the most susceptible. Of course, some radioactivity is naturally occurring, but humans concentrate this radioactivity through mining and use of oil, gas, and uranium fuels. They create new radioactive isotopes never before seen in nature by a number of industrial processes, and they do this in order to produce electricity from nuclear reactors and also to produce atomic weaponry. Further, humans release these radioisotopes to the environment through processes like mining, milling, and enrichment of uranium, fabrication of uranium fuels, and fissioning of those reactor fuel rods. And I would point out one more industrial process, which is reprocessing. That's the chemical separation of uranium and plutonium contained in irradiated reactor fuel that has been removed from the core. You then have to store the highly radioactive used up fuel or radioactive waste, some of which can last for thousands of years. And all along this process, wastes are released and environments are contaminated and people are exposed. So for an accessible explanation of the uranium fuel chain, please see Beyond Nuclear's handbook pictured here. The link is included on the slide at the end of this presentation, which contains a list of references. So let's go through some technical basics first. Ionizing radiation is expelled from the nucleus of an unstable or radioactive atom. This expulsion, known as decay, is represented by a unit called a becquerel. The 1 becquerel is equal to 1 atomic disintegration per second. Different radionuclides have different numbers of decay per second and give off different kinds of radiation. Each type of radiation can be stopped or travel through different types of material depending on the density of the material and the quality of the radiation itself. The first 2 types of radiation here, alpha and beta particles, for instance, are less damaging outside our bodies, where they can be blocked by paper or aluminum. But they become more damaging if the isotope that is ejecting them is inhaled or ingested, since there's nothing inside our cells like paper or aluminum to block their energy. In this case, each one of these disintegrations or hits, represented by a becquerel, may cause damage and disease. So when we think of radioactive releases from nuclear reactors, we often think of catastrophic releases like Chernobyl or Fukushima. But reactors have huge unplanned releases, which are not the result of a meltdown. And they can continue to operate after these releases. For instance, in 1979, Oyster Creek in New Jersey released 1 million curies, which is a large release, but it wasn't termed a disaster. And then there are the releases that are allowed that fit in with the official idea of a permissible dose. But permissible does not mean safe. And we know this because this dose was originally called an acceptable injury limit. Radionuclides released from these different types of circumstances can be different. With this in mind, this slide offers both a partial list of radionuclides released from a catastrophic meltdown, this would be Chernobyl, and also a partial list of radionuclides released routinely. I also include here nuclides like uranium because civilian reactors use uranium as fuel, and uranium is almost always on lands of First Nations or those who live close to the land. So it is important to weave uranium and its impact into any presentation on the health dangers of nuclear power. The orange arrows designate the radionuclides we often focus on to determine health outcomes from exposures, but these are not the only ones of concern, however. For instance, this blue arrow points to xenon-133, which is fat-loving and could actually be responsible for a substantial dose during the first days of a catastrophic release. And please note that radium and radon are starred because they and their decay products can be relevant to health impacts of oil and gas technology. 4 main things can happen to a single cell once it's hit by any type of ionizing radiation. The cell can be undamaged or damaged and properly repair itself. The cell can be so damaged that it dies. The cell can be damaged, improperly repair, live and reproduce, but the daughter cells die. Or the cell can look repaired, but hold mutations that it passes on to the daughter cells, which in turn are mutated and survive. For lower doses especially, this is the important process because it can result in diseases that can show up in humans and other animals decades or even generations after the initial damage. And how much damage a hit by radiation will cause is variable due to many factors, including the age of the person, the difference between people's ability to repair themselves, the difference between certain types of cells, the type of radiation, and the type of exposure. So there are many different types of cells within the individual that perform specific functions. For instance, brain cells, they're different from nerve cells, which are different from heart muscle cells, and so on. Some cells, like bone marrow cells, are more sensitive to radiation than other cells. Finally, studies show that children, women, and pregnancy are more susceptible to the effects of radiation. That means they're more likely to get disease from exposure to it than the male standard used as the basis for our radiation exposure standards. And these impacts are my focus. To that end, let's look at where the radioisotopes that were listed previously travel and collect in a pregnant body. I should note that xenon-133 is not included in this list, but remember, it's fat-loving, so it will go to fat cells. What I would point out from this graphic is the collection in the fetus of nearly every radionuclide on this list. And do note, radioactive carbon and radioactive hydrogen, called tritium, can collect in fetal tissue at twice the concentration as they do in maternal tissue. They are the basic building blocks of the human body, as is calcium, which is mimicked by radioactive strontium, which is a bone seeker that lodges in the mineral bone of growing skeletons and likely will not leave the body for most of that person's life. So, how can we determine how damaging a specific radioisotope is for any given organ? Well, we need to convert from how radioactive something is in becquerel to the kind of damage that it causes in a unit called sieverts. And this is the formula that supposedly does that. Now, this is their basic attempt to calculate and to help us understand how much and what kind of biological damage comes from exposure to different kinds of radioisotopes and the kind of radiation they give off. This formula encompasses individual nuclides that are deposited in the body, so this formula would be for an internal dose. There are also biokinetic models describing basically where in the body the radionuclide goes and when or if it leaves. But the basis for these models has mostly been higher doses and mostly representative of a male adult. For low-dose dosimetry and chronic exposure, these models are lacking because they are avoiding the intricacies and individualities of a fully functioning human system. For instance, children are represented as little adults, and damage to children's organs is almost based exclusively on weight, not function or dysfunction over time. Adults are represented as slabs of meat. That's my characterization because these models are not reflecting the complexities of the damage, such as cumulative damage by continuing exposure. That's not accounted for. Neither are the metabolic or hormone pathways, particularly the estrogenic pathway, which seems susceptible to action by ionizing radiation. Biokinetic models are not sex-specific. A male model is still used for females, and they are also not age-dependent. In general, these formulas represent transport systems, but there are many more systems in our bodies, one of which is the reproductive system. So basically, research studies use a dose to a non-pregnant uterus to determine pregnancy damage. Damage to a number of processes during pregnancy are either poorly accounted for or not accounted for at all, such as damage to sites in stem cells responsible for blood cell formation, the maternal exchange system, and damage to embryo organ tissues and placental function. Although they are in the process of correcting for some of these shortcomings, we unfortunately have a whole body of studies that have made poor assumptions about dose impacts, particularly from recurring lower doses. And unfortunately, these studies have scooped the narrative about what the damage is or is expected to be without coming clean on the depth and the importance of the uncertainties. So there are a lot of uncertainties and poor assumptions used for formulas for dose reconstruction and representation of damage. The mantra of the mostly harmless low dose, along with other really poor assumptions, are routinely used to dismiss radiation's damage, leaving no identified explanation often for the studies that find disease increases, when in reality, radiation could be responsible. So official studies of 3 Mile Island, Chernobyl, and Fukushima all suffer from poor assumptions. Underlying all of this uncertainty is how low is a low dose? So this is an autoradiograph of a plutonium particle in the lung. Now, determining what is a low dose is an epistemic problem that extends well beyond plutonium, but I'm using plutonium as an example. And this is particularly important for radionuclides that are internally deposited. So you can get a very small dose when you divide this plutonium particle dose among all of the lung tissue. But as you can see by the starlight deposition of the energy, the dose isn't being delivered to the entire lung. So what is this dose? Well, there is research and publication that's currently examining this. This 40 micron region around the particle is about 2/5 as wide as a strand of hair. And the dose per day that this cell is getting is 1,000 to 10,000 gray. For a comparison, a whole body dose delivered to a human being of 1,000 gray causes almost immediate unconsciousness and death within an hour. So clearly that means death for this cell. But what we also know is that the cells surrounding the cell can also be affected by the dose, even though they were not directly exposed. That's a phenomenon called the bystander effect. So again, I ask, how low is low? And what happens with a radionuclide like strontium, which we talked about before, which is a bone seeker, which goes into a growing skeleton and doesn't leave for most of that person's life? That's why it's important that we know what the studies are actually showing. So continuing exposure to radiation levels at or near natural background doses are associated with increases in childhood cancer, primarily leukemia, but also central nervous system cancers. You can see that represented here on this slide by these two graphs. For reference, naturally occurring background radiation exposure is an average of 80 to 100 millirem per year. And note that the 225 nanosieverts per hour is about 197 millirem per year, which at which the exposed individual would have twice the chance of developing the disease. These dose levels are also associated with subclinical impacts leading to behavioral problems and underperformance in school, particularly in math. Dozens of studies show increases of childhood leukemia around normally operating nuclear facilities, particularly in children under 5 years of age. There were increases of childhood leukemia in children who resided within 5 kilometers downwind of the Kreml boiling water reactor in Germany, represented here by this map. Following this study and others like it, the German government funded a countrywide study which showed that children under 5 years of age living within 5 kilometers of reactors in Germany had elevated rates of leukemia. A cascade of studies followed in other European countries showing much the same result, and you can see this data pooled here in this table, which was compiled by Alfred Körblein and Ian Fairley in Europe. We have independent studies in the US showing increases in disease as well, but we have yet to have an official US-wide study undertaken on this critical issue of childhood cancers around nuclear facilities. The most recent study on perinatal mortality in Japan following the Fukushima meltdown and explosion in March 2011 shows that during 2012 to 2019 time period, there were 590 excess perinatal deaths in the area shown here in both dark and light gray. Further, there were, quote, massive increases in 2018 and 2019 in the 4 core prefectures, end quote. Those are represented here on the map in dark gray, meaning the issue continues past the initial plume exposures and could point to ongoing radioactive contamination as partly responsible. The study recommends, quote, continued investigation of perinatal mortality in contaminated regions of Japan. After living with Chernobyl contamination for decades, studies indicate radiation damage may be carried and compounded across generations, and a recent study in the Bryansk region of Russia, inside the green circle, associates birth defects with living in contaminated areas. Some of these birth defects are de novo, meaning that they are appearing in this population for the first time. Further, study projections indicate that certain birth defects will continue to increase in the next few years. But it's not just radioactivity from nuclear sites. Complex environmental exposures from a variety of sources, such as chemicals, infectious agents, and lifestyle factors, and how they can affect a person's health, is called the exposome. The study by Korsakov that I mentioned on the last slide in the Bryansk region of Russia uniquely also examined and demonstrated additive and potentially synergistic impacts in areas contaminated by Chernobyl radioactivity and industrial chemicals both. Exposome investigations need to lead the way into further research on radiation damage, as do what I call biomarker studies that I did not mention here. Biomarker studies look at the traces radiation leaves in our cells to determine the damage. And that's the end of my presentation. I will be happy to take any questions, and this is a list of resources that I used to compile my talk.